The emerging fungal pathogen Candida auris has lineages that behave differently, which may complicate treatment and infectious control of this microorganism. Welcome to Microbial Minutes, ASM's update on what's hot in the microbial sciences, Tricky Fungus Edition. I'm Julie Wolf, and today's paper comes from the Journal of Clinical Microbiology, which shows that differently behaving lineages may complicate the emerging fungal pathogen Candida auris. So when I say emerging, I mean very recently. Candida auris was first patient in Japan in 2009. Since then, it has caused outbreaks on almost every continent that has human inhabitants, and it's especially associated with hospital-associated outbreaks, so outbreaks that occur within healthcare facilities. And these are dangerous for a number of different reasons. Number one, patients often have an underlying condition that makes them particularly susceptible to this infection. Number two, the disease looks very similar to other infectious Candida species and can sometimes be misdiagnosed. And that can be bad because number three, Candida auris has intrinsic antifungal susceptibility. And so it's going to require slightly different therapy or therapeutic effective doses of antifungals in order to treat the infection. Now, this study comes from the United Kingdom, where there are at least 3 lineages that have been reported. Those come from various parts of the world. They're named after where they were found. So these are the South Asian, South African, and Japanese Korean lineages. And these lineages have already been demonstrated to have different virulence capabilities within a wax moth model of infection. And so what this trio of researchers wanted to ask was whether these lineages also vary in other important phenotypes. As we'll see on the next slide, they looked at a number of different characteristics. They looked at the ability to form pseudohyphae, which is a different cell shape. So these fungi can grow either as a yeast, which is a round shape, or a pseudohyphae, which is an elongated cell shape. They looked at the ability of these different lineages to cause aggregates or to form big clumps of cells, which has been shown to protect against, to have a potentially protective phenotype against both immune onslaught or antifungal agents. And they also looked at the ability of these different lineages, also called clades, as you can see in the table, to grow in the presence of actidione, which is another name for cyclohexamide, a compound that has some anti-yeast capabilities. And you can see those growth capabilities in the figure on the right-hand side. The labels of 1, 2, 3, and 4 correspond with 2 different South Asian clades, a South African clade and a Japanese Korean clade. These are different isolates from the hospital where the work was done. And that all of the strains can, of course, grow on a rich medium, but when actidione is added, the South Asian clades are no longer able to grow. You can see from the chart that there are different abilities of these different clades to either form pseudohyphae or aggregates as well. And so that suggests, or demonstrates, I should say, that these phenotypes differ on standard mycological media, which will have important implications, as we'll see on the next slide. This is going to be important because the ability to identify Candida the CDC is already concerned about. They offer very helpful guidelines on their Candida auris page, and in fact also offer help through their antimicrobial resistance lab network. And we'll link to some CDC information down below. But the ability to rapidly distinguish clades has epidemiological and infection control utility, especially where one thinks in different hospitals where multiple clades may be circulating. It's important to identify what those clades are or those lineages are and how they can be treated. It's possible, based on these findings, that the growth on actidione may facilitate differentiating some of these different clades. But whether these different in vitro phenotypes that have been demonstrated by this group correlate to the ability to cause an outbreak is something that has yet to be figured out. There have been a number of different pieces about Candida auris, and we'll add links to those as well. One of those is the history of the discovery of the microbe itself, which was written up in Wired, where the chief of mycotic diseases at CDC, Tom Chiller, negatively compared Candida auris to other diseases such as Ebola, saying this bug is the most difficult we've ever seen. It's very well written, and I recommend reading it. This is also one of the top 5 infectious disease concerns to watch in 2019, one of the reasons that we've highlighted it here on Microbial Minutes. In summary, different Candida auris lineages have different phenotypes and antifungal susceptibilities, and this may cause problems down the line. I'd like to thank you for listening, and thank Ray Ortega for production. I'm Julie Wolf, and I'll be with you next time on Microbial Minutes.